My name is Matthew Adams. I am the Associate Director of Graduate Admissions for the Helmholtz College of Arts and Sciences. I just want to thank everybody for coming today. I want obviously appreciate you guys taking your time to come out and hear from some of our great faculty members. Some of you have attended in the past some of our virtual lecture series. We do these in the fall and in the spring as well. So I'd like to introduce today's presenter, Dr. Andrew Ozga. Andrew is a NSU professor in the Department of Biological Sciences here at Nova Southeastern University. He's broadly interested in host genetic and microbial variation across human populations and non-human primate species. His current research includes an effort to understand the impact that host genetics and oral health on oral microbial communities within South Florida residents. He has written and contributed various publications, including journal articles, edited collections, and books. He has also presented in a wide range of subject areas, such as non-invasive genomics and Gombe chimpanzees, microbial genomes, oral microbiome variations, comparison aDNA, and ancient dental calculus. So Andrew, jump in after and correct me on any of those if I'm off. But today's presentation is really, in large part, an informational webinar regarding ancient DNA and oral microbiome diversity in human and non-human primate populations. But at this point in time, I'd like to turn it over to Andrew, who will be available for questions with my assistant director, Nicole Chavez, after the presentation, especially if you're interested in our computational molecular biology graduate certificate. It's 12 credits. We also have a Master's of Science in Biological Sciences in a research focus concentration within that program as well, and it's a 36 credit program. So Andrew, take it away, and we want to thank you again for putting this on.
Great. Thank you both, Matt and Nicole, for putting this together and inviting me, and thank all of you for checking out the research that I'm going to be talking about. Okay, let me get started here. Okay, so as Matt mentioned, my name is Dr. Andrew Ozga. I am an assistant professor here at Nova Southeastern. I just started last year, and it's been absolutely awesome so far. The faculty, students, everything's been really phenomenal. So I'm really excited to be here, excited to be talking to all you folks about this stuff. So I titled this, kind of a title, I guess, Human and Non-Human Primates Genetics, Genomics, and Microbiomes. And we're going to kind of walk through some of that stuff a little later, but I'll start out by telling you a little bit about myself, some of the stuff that I'm doing here at NSU in regards to teaching and research, and then I'll kind of dive more into my stuff, and then I'd be happy to take some questions if you have any at the end. Hopefully I don't run into any tech or problems or anything like that, but yeah, we shall see, hopefully not. Okay, so I am originally from the west suburbs of Chicago, and I've bumped around to quite a few different colleges over the years. I always really had a passion for biology, even as a kid. I remember in middle school and high school really liking just some of the basic concepts of evolution and just how cells were structured. So I went to do my undergrad at Marquette University in Milwaukee. And it was there that I actually got a little bit more focused into the skeletal system. So before I was into microbiomes and genetics, I started out by being into bones. So I was basically working with a really prominent osteologist at Marquette, and he had a collection of about 1,000 or so skeletal remains from a pauper cemetery in and around Milwaukee from around the turn of the century. So basically 1800s going into 1900s. So basically I started examining that and looking for different paleopathological components. So I got really interested in the different types of arthritis that impact and can appear on the skeletal system long after someone has passed away. So because I worked on that in some of my undergrad work, I decided to write my master's on that topic. Basically, I was looking specifically at diffuse idiopathic skeletal hyperostosis, which is also known as DISH. It's basically a fusion of the anterolateral ligament of the spine, and it is pretty debilitating, and it was really prevalent in this pauper population, strangely enough, even though it's supposed to be associated with more affluent, diet hardy populations. So I went to Ball State and finished up that master's there, so Ball State in Muncie, Indiana. And then I went on over to University of Oklahoma. I basically touched base with a bunch of different professors and told them I was interested in genetics, and Oklahoma offered me basically to take me on as a research assistant and as a TA and all that kind of good stuff. So that's where I really started getting into the genetics field. I was basically dealing with a lot of different types of projects there, but one of the ones that I became really passionate about was profiling the different types of bacteria in underrepresented communities. So a lot of the bacteria information we know about bacteria of the human body come from European-derived populations. And one of the benefits of working in Oklahoma is that there are many, many Native American tribes there, and they were willing to work with us. And basically, we were able to sample their saliva and their feces to get a really good glimpse at to basically what's happening in native indigenous peoples in North America in regards to their microbiomes. So in addition to that, I was also working in the field of ancient DNA, which brought me to my postdoc research, where I went to Arizona State. I relocated to Phoenix, and I was working with the Institute of Human Origins, which has some of the more prominent researchers in the field of evolutionary biology. And then I took another postdoc there in the Center for Evolution and Medicine, which kind of ties together a lot of different medical and evolutionary concepts, and that kind of spurred me to in a couple of different directions. So I've been growing out my quarantine here, so that's me at 18. I look a little similar to that now. And as I mentioned, I was looking at the spines of a lot of these individuals. And over the years, I've really gotten the opportunity to go to some pretty amazing places. My dissertation was actually on microbes and viruses in the guts of not only Native North Americans, but Native South Americans. So this is a picture of me on the Pisque River in Peru, in the jungle region of Peru. So we were able to basically make kind of contact with those communities and explain to them what we were interested in doing, and kind of continue to follow up with them about their concerns. And eventually we were able to get a really nice paper out of that examining diet and the gut microbiome of native Peruvians. So all of that has kind of brought me here. I've jumped around to a lot of different states, but I've always really enjoyed teaching. And before I talk about my research, I just wanted to mention a couple of classes that I teach here at NSU. So I've taught biology. I taught biology at a community college for 6 years during my Ph.D. And basically, one of the things that I really do enjoy is being kind of the first contact, really getting to chat with those early freshmen that are really interested and really amped up about biology in general biology. So this really is a class that I do enjoy teaching, because there are a lot of really passionate students there. They're kind of getting used to the whole idea of studying in college. So I'm kind of happy to help out any way I can with that process. And we talk about basically different types of evolution. You see here, these are a couple of different, a single mouse species, but with a couple of different phenotypes, physical traits there, because they have been basically naturally selected to be either brown or white, depending on the environment in which they live. And we cover basically everything in that course, some of the basic chemistry, chromosomes, that's a ribosome up there in the top that helps cells produce proteins, and of course mitosis, everybody's favorite topic. So that's a course that I usually teach a few times a year. I'll be teaching 4 classes of general biology this coming winter. One of the other courses that I teach here is microbiology. So this is actually my first time teaching microbiology at NSU is this semester, and it's really been absolutely awesome. I also teach the laboratory component of that class too. As you would suspect, microbiology is really all about the different types of microorganisms that inhabit the planet and inhabit us. We cover a wide range of topics, basically function and dispersal of all these different bacteria. But this also includes microorganisms too. So we talk about fungi, we talk about parasites. And then the really neat thing about the class is it has that laboratory component, and what students do is they will take an unknown bacteria. So basically the first couple of weeks of class, we will have them basically sample some soil from their whatever location they choose. And then that soil is then used, we grow out some bacteria from that soil that they get, and then they're working with the same bacteria week after week and putting it through a barrage of different tests to find out what that unknown bacteria is. This past week, we just actually used some of that bacteria to examine antibiotic resistance to different types of antibiotics. We figure out if they're fermenters of any kind. We figure out if they basically can break down nucleotides or a whole other variety of things. So that's one of the classes I teach here. And then lastly, I also teach a graduate class called Introduction to Bioinformatics. So in addition to working in the lab, I'm also a bit of a computational biologist. Basically what that means is that you're dealing with a whole set of, basically all it is A's, C's, T's, and G's. So those are the three nucleotides that make up DNA. And what a bioinformatician's job is to do is basically to make sense of a lot of that information that comes from the sequencing machine. So you can have a bunch of great wet lab skills, but if you don't really know what you're doing once you get the data back, that's a kind of an issue. So what we do with this class is basically we're dealing with a whole variety of tools. And I walk through with the students how to understand command line coding. We do a little bit of Perl and Python scripting. And then we're also using some cloud databases through Galaxy. And we got a small grant to do a bit of cloud computing through the Pittsburgh supercomputing cluster. So basically showing how you deal with really big, you're talking about like trillions of bases of sequences. How do you make sense of all that stuff? So that's a really fun class to teach, and I've gotten really good feedback on it, and that's part of the computational biology certificate program as well. And I just wanted to mention one last thing with the courses. So I am actually in the process of creating a new course. So one of the things that I'm passionate about, I'm actually kind of a classically trained anthropologist, even though I am a biologist as well. So I am putting basically a course together that I have designed associated with biological anthropology. So basically, what makes us human? What makes primates the way that they are? Why do they differ? Why do they differ from a lot of other animals in the animal kingdom? So we're going to be touching on a bunch of really cool topics. That'll probably be in fall of 2021 or winter of 2022 if the course goes through, which I hope it will. So yeah, as I mentioned too, I do a little bit of research in addition to teaching. And what I'm currently doing with a couple of undergrad students right now and a series of pre-dental students over in the College of Dental Medicine is we are basically looking at the different types of bacteria in the mouths of South Floridians. I will mention this a little bit later as kind of part of my research, but I wanted to just talk about how up top I am basically working with and helping to train undergrad students. I don't have any grad students yet, but I hope to get some that are basically kind of interested in the field of microbiomes and of basically what these bacteria do within human populations. All right, so this kind of brings me to my research. So I'm going to talk for a little bit about a few different topics. And the first is going to be ancient DNA. These are kind of tied together a little bit, but I'll kind of say, where they differ a little bit. And then I'll talk about non-human primates. And then I'll talk about some of the research that I've been doing over the course of my career with living human populations. Okay, so basically ancient DNA is the study of really, really old genetic material. And the reason this is important is because there's been new technology recently that has come out that have allowed us to look back in time further than we ever thought possible. And we actually do this through a series of different biological specimens. So we can use bones, we can actually use soil to examine ancient genomes. We can look at feathers from museums. We can look at basically any soft tissue or skins that have been preserved. And we can look at dental calculus, and you can see basically up on those top two pictures, that little kind of crusty stuff that you see on the teeth, that is dental calculus. That is the fossilized or technically calcified form of dental plaque. Everybody has dental plaque. I mean, if you run your tongue across your teeth, you'll probably feel some right now. And that's basically bacteria laying down a little biofilm on your teeth. And that biofilm over time can harden, and it has a multitude of uses within it. So I've actually used some of my career to investigate and kind of explore what this dental calculus, what it can tell us and where it's found. It's considered to be ubiquitous, meaning that it's pretty much present across all mammal fossils. So we see calculus, potentially hundreds of thousands of years old, and it contains within it basically some genetic material. Sometimes you get things called phytoliths, which are basically just little remnants of seeds and potentially some natural agents that they've eaten that got kind of stuck in the plaque. You see viruses, you see bacteria, and you actually see a lot of information about the host genome itself. So some of the research that I've been doing in the field of ancient DNA has been on dental plaque, not all of it. What you're seeing there in the bottom right is a project that I was working on in South Africa, dealing with between 50,000 and 400,000-year-old bovid remnants. So basically hooved animals in South Africa. And we were trying to understand the genomes from those organisms. And the thing with ancient DNA is that you never really know if it's going to work. So I've had a lot of really cool projects. Some have not panned out. That one in South Africa did not work, unfortunately. But it's, it's always fun when it actually does work. So this is the field of ancient DNA. Even though we don't have a laboratory here at NOVA to do ancient DNA research, I have a lot of great colleagues all across the world, and I've been privileged to, basically they have opened up their labs to me, and they say, if you ever have any new projects, feel free to come back. So I still plan to work in the field of ancient DNA, even though the lab work won't be done here. So this is a picture of me with a 16,000-year-old dire wolf jawbone. This is a really nicely preserved specimen. You can see it there. This is the binomial Latin name for this is Canis dirus. And yes, dire wolves were actually a real thing. They're not just made up from Game of Thrones. They existed in America between about 50,000 and 100,000 years ago. And we actually have a paper right now that's going through in Nature, and it is going to be examining the genomes of some of these ancient wolf kind of species. So unfortunately, even though that jawbone looks amazing, and I had to destroy it to get DNA out of it, I was not able to recover DNA from that sample, but we were lucky enough to have a few other dire wolf samples from the Northwest that did pan out. So it's just kind of one example of the different types of ancient DNA research that you could have. So I wanted to just kind of take a step back and explain what makes ancient DNA, because that's actually been part of my research agenda, is trying to figure out exactly what's going on within the genetic material of these different samples, whether they be teeth or calculus or even hair. So basically DNA is made up of 4 bases, A's, C's, T's, and G's, adenines, thymines, cytosines, and guanines. And they are made up into a nice kind of double helical nature. So once an individual passes away, human or mammal or whatever, basically their DNA starts to degrade within their, and basically after all the soft tissue goes away, you have skeletal remains, and depending on how old they are and depending on where they are located in an archaeological context, they may actually preserve DNA pretty well, or they may not preserve DNA very well. You want to have basically a cool, dry area. Water damages DNA within bone. And basically DNA has only a half-life of, I think, a couple hundred years. And basically what that means is after 100 years, you get half of all the genetic material is gone. And then after that same amount of time, you get another half. So we can't really go back that far to look at DNA. It basically degrades in a way that we kind of understand, which I'll talk about in a second. But the oldest genome that's ever been reconstructed has been about 800,000 years old from a horse population in Asia. So we can go back pretty far to basically understand what is the genetic material of these different organisms and what can it tell us about evolution. How have these horse populations, how have these human populations changed over time? And that's a lot of the research that's being done in the field of ancient DNA right now. How did we get here? How did humans populations converge and move across out of Africa and across Europe and Asia and into the Americas? So one of the things that we basically understand about DNA is that it gets damaged. So, basically, if you're able to take a saliva sample from somebody now, chances are that the DNA is going to be great. You're going to be able to recover some of the, if you're looking at the whole genome, it's about 3 billion base pairs. If you're just looking at the mitochondrial genome, that's only about 16,000 bases. But basically what happens is that the bonds holding the nucleotides together, like we saw, basically the rungs of that double helix, they basically start to fragment. So the bases on the same strand basically start to kind of fragment. So this is known as depurination. And this is one of the issues with ancient DNA studies, is that you start getting a bunch of fragmented pieces. As I mentioned with the bioinformatics class, one of the things that you can do with bioinformatics is determine how much of your DNA is fragmented. And that actually gives you a hint as to whether your sample is contaminated by modern DNA. So I've worked with a lot of different populations, a lot of different types of samples and a lot of different human and non-human populations. And basically one of the big things that you have to be concerned with in ancient DNA research, that's why I had that hazmat suit on in the other picture, is contaminating the sample with your own DNA. So basically seeing DNA damage is a good thing, and it actually tells us that the DNA is legitimate and it is old enough to look at. And there's another process known as deamination, which is the removal of an amine group from a cysteine, which turns it into a uracil. And basically what that means is that it's basically turning into what we see with RNA, with basically single-stranded RNA. So the reason I mentioned these two topics up top here is because you can actually bioinformatically go in and take a look at sequences and observe these changes and then account for that. There's also methods in the lab to fix these and repair them. So what some laboratories will do with really old samples is they will sequence some DNA from, say, a human bone from about 100,000, we'll say 30,000 years ago, probably get some DNA from that. And then they'll sequence it with a repair mechanism that fixes all of these cuts in the little double helix. So you compare those two together and you can actually see where that damage occurred and how you fixed it. So the reason I mention this is because I've had a fair number of publications that have really kind of dove into what happens with ancient DNA, what makes it ancient. That top paper up there, Differential Preservation of Endogenous Human and Microbial DNA in Dental Calculus and Dentin, what we did is we took a series of different samples from all across the world that were paired with both calculus and dentin. And we sequenced the dentin side. We sequenced just the actual tooth root itself to get DNA from that. And then we sequenced the DNA calculus, and we observed the different changes between those two genomes. So what you see is that there is even though you probably get more DNA from dental calculus, you get a lot of bacterial DNA too, but the DNA that's preserved within the human root is really nicely put together. You see more fragmentation within dental calculus because you actually have a lot of bacteria in there, and those bacteria can release enzymes that basically break down the DNA at a more rapid rate. I was also part of a couple of projects looking at different extraction methods from skeletal remains in tropical environments, which are notoriously difficult to deal with. Water is not a friend of the ancient DNA community. Basically, it breaks apart DNA at a more rapid rate. We also basically looked at what not only the mitochondrial DNA, which is the smaller genome within humans, but also the larger nuclear genome. So we were looking at the efficacy of whole genome sequencing capture on ancient calculus and dentin, similar to that other paper. And then we also looked at some of the differences between sequencing methods within these different ancient bacteria studies. So the way you look at the microbiome, there's a few different ways, but the way you look at bacteria that are present in dental calculus of old individuals, whether they be humans or whatever, is you're basically looking for a gene known as 16S. This is a ribosomal DNA sequence. And it's really nice because you can actually sequence a very small fragment of DNA and get a really good glimpse at the taxonomic information of an entire ecosystem. So basically 16S has allowed us to pretty cheaply look at all of the different types of bacteria, even though it's just a very small DNA fragment, all bacteria have that gene and most archaea. So it allows us to really kind of look at a bunch of different things. But what we found is that depending on the region of the 16S gene that you look at, you're going to be biasing your samples in one way or another. There's certain segments of the 16S gene that when people look at those, they give you a different readout on what bacteria are there than another segment within that gene. So that was the purpose of that paper in the bottom right, and these have been pretty well received within the scientific community, especially within the ancient DNA community, where we're all kind of just working together to figure out exactly what's going on. Okay, so one of the other projects that I've been really passionate about is some of the work that is basically coming out of the chimpanzee populations within Gombe National Park. Jane Goodall has always been a personal hero of mine. In 1960, she went with her mom over to basically that little arrow, what you're seeing there. It wasn't a national park at the time. It was just called Gombe Stream. It's tucked right up against Lake Tanganyika in Tanzania. And basically, she was interested in looking at chimpanzees. Prior to this time that she went there, chimpanzees were considered to be brutish and have absolutely no emotion. They were just kind of evil. People didn't really know what chimpanzees did or how human-like they were. So based on her observations, she was able to basically kind of change how the entire world looks at this species of organism. I've been really lucky to be involved with some people basically to help run that conservation area now. It's pretty small, but it contains about 200 chimpanzees. I think we're just talking about a matter of probably 20 or 40 square miles. Unfortunately, I have not attended, have not gone to Gombe yet, but hopefully one day. But what actually kind of led me to this research path is some of my collaborators said we have some of the bones of some of the chimpanzees that Jane Goodall had studied in the 60s. Do you want to take a look at some of their dental calculus? So obviously, I jumped on that possibility of researching something so iconic, at least to me. And what we've done from that is there's some papers that are still in development right now, but I'll kind of just talk about some of the results that we've seen from these population of chimpanzees. So this kind of spurred on my interest, this opportunity spurred on my interest in the field of non-human primates. So basically I'm interested in gorillas and orangutans and chimpanzees and how their genomes and how their bacteria, especially in their mouths, differ from what we see in human populations. So one of the papers that came out of this was something that I published last year in Scientific Reports. And basically it was examining the shared bacterial types between human populations and chimpanzee populations. And basically, so what we're seeing here is we're seeing a group of a whole series of ancient DNA. So this is a plot that actually tells you the relationship, the shared relationship between bacteria. So blue there represents humans. So each of those dots is a single individual sample from a single individual. And then each of those red dots is a sample from chimpanzees from Gombe National Park. We also included some Neanderthals there. So those orange dots are representing 4 different Neanderthals. So basically what this study showed is that there is a much more similar bacterial profile within chimpanzees than within humans. So every dot that you see here, the closer the dots are to one another, the more bacteria types they share. So as you can see, Neanderthals are kind of spread out there. There's a couple of human populations that are a little spread out, but the core difference, you see that the red and the blue dots are in separate areas. So this basically means that the bacteria types that you're seeing in humans are not always reflected in chimpanzees. Now, this kind of makes sense because obviously chimps don't have dental care, but this was actually a lot of these human samples were from 10, 20, 30,000 years ago, so they didn't have dental care either. So it could be a result of their the host genome impacting the type of bacteria in the mouth. It could be the diet that they're consuming. So I didn't try to posit exactly what's driving these changes, but what we're also seeing too is a let's see here. So hold on, frozen, there we go, okay. What we're also seeing is there's an increased abundance of bacteria that we know to be pathogenic within these different species. So what you're seeing here is a bar plot that's showing the percent abundance of the total bacteria examined within each of these samples. And the Porphyromonas is basically a common bacteria known to cause cavities and gingivitis. We actually see this super abundantly in Gombe chimpanzees compared to historic human populations. So that's probably because I guess chimpanzees are getting, basically they could be getting more dental related issues than human populations. Anyway, there's kind of, this is pretty tough to know exactly what's driving this, but so you are seeing some differences in known pathogenic bacteria between these two groups. And then additionally, I have an ongoing project that's looking at the basically the dental calculus from great apes and museum collections. So I went through and surveyed a bunch of dental calculus from gorillas, chimpanzees, and orangutans from different museums. And that is a project that I'm continuing to this day. And we hope to kind of try to figure out exactly what types of bacteria are more common in non-human primates than human populations. Okay, so one of the other things that I've been working on in regards to Gombe chimpanzees is we actually had a lot of samples of urine from Gombe, which is kind of strange because it's kind of tough to get urine from a wild animal population. So basically we had some really great field hands that we worked with, and we were interested in taking a look at comparing different biological sources and how that impacts the presence of basically the host genome. And in order to collect that urine, we actually had field hands use these tarps and basically sit under the chimpanzee nests when they woke up and kind of hold up the tarps and collect the urine into a sterile container. It was probably not the greatest job in the world, but we're grateful to them that they're willing to do that. And we also looked at feces, because feces has been a long studied non-invasively collected sample. By that I mean you cannot touch these chimpanzees. They are endangered. They're critically endangered. So we can't get anywhere near them, but we want to understand their genomics. So for a while, feces was used. However, the drawback of feces is that it contains a lot of different enzymes that break down DNA really rapidly. So what we did is we collected urine, we collected feces from living chimpanzees, and then we compared that to dentin and calculus from deceased chimpanzees to see, and we used the exact same kind of sequencing methods and extraction methods on each of those in an attempt to see how much genome can you recover from each of these different sources. And what we found is that urine is actually a really amazing source of non-invasively collected genomic information. Feces was really not that great either. I was going to make a bad pun, but I'm going to refrain from that. And both of those did quite better than dentin and calculus. So this is kind of another methodological paper, but it's an important one because we weren't able to, when you really want to study and understand something's genetics and genomics, their entire genome itself, and with critically and endangered populations, you can't touch them. So we've been trying to find new ways to examine these, and I think this prospect that urine is a really great resource is going to be really helpful to individuals in the animal genomics world that haven't basically used it before. So this was that paper, and that actually just came out in October of this year. So we're really pretty proud of that one too. Okay, so that basically brings me to my final component here is the modern oral microbiome. So I really like doing ancient DNA work. It's frustrating as it could be. I really like working with primates, but don't really have access to Gombe chimpanzees here in Florida. There's a few primate reserves, but still. But one thing that I'm really passionate about is basically understanding and helping put together the pieces of what we know to be the human microbiome, particularly the oral microbiome. This top figure is a paper that I wrote a few years ago. Basically, we got 5 different cities of the Cheyenne-Arapaho indigenous American nation to participate, and we examined their oral microbiota compared to non-indigenous people at University of Oklahoma. And we found a stark difference between these two populations. There was a few bacteria that were much more abundant in these native groups than non-native groups. So one of the things that I've been passionate about is basically helping put together a larger database of populations that have been ignored by the microbiome literature. And one thing I'm really excited about down here is that South Florida is a really diverse place. And one of the projects that I got a little bit of funding to start down here, and we're actually doing sample collecting starting in a couple of weeks. But basically, I am just trying to investigate and see if there is any sort of association between the types of bacteria in the mouths of South Floridians and some basic demographic information like ethnicity and sex and age and all that kind of thing, but also their dental profiles. So I'm working with some periodontitis and some dentists that are going to be collecting a plaque sample from these individuals and basically trying to, they're going to do a little dental workup, and we're going to try to see if we can connect any dots as to the presence of these different bacteria in a really diverse population and how that may correlate with demographic information and oral health information. So they'll be taking periodontal pocket depth, plaque analyses, all that kinds of things in the clinic when the samples are taken. So that's basically the project that I'm continuing with now. And I think it's really important because plaque can tell us a lot of stuff. As I mentioned, I've dealt with fossilized plaque in the past, but I also like dealing with plaque in living individuals too, because the oral cavity and the bacteria within the oral cavity really have a lot to tell us about a lot of more systemic issues within the body. There have been links with the oral microbiome to obesity, HIV, and different viral presence impacts the oral microbiome. Alzheimer's disease has been associated with upticks in abundance of different types of bacteria, things like liver cirrhosis, IBD, all these other things. So the oral microbiome really has I think a lot to tell us, and I'm really interested in exploring the different types of basically oral diversity within a population like what we see in South Florida that is itself so diverse. So basically, yeah, the kind of the main goal of a lot of my research has been to, within the ancient DNA field now, has kind of been to understand the evolution of pathogenic and commensal bacteria across primate and human populations. And then now with current living populations, kind of associate oral bacterial load and diversity to basically what this means about the host. How is the host kind of impacted by the presence of these? And then kind of overall too with ancient DNA, what can it tell us about evolution? So one project that I'm working on right now in ancient DNA is we're looking at basically the evolution of wild turkeys. So I have a lot of basically kind of, things that I'm interested in, but these I would probably say kind of best describe everything that I've kind of been working for the last 10 years or so. So anyway, with that, I will end it. I really appreciate all of you for sticking around to listen to me, and I think we can take some questions. I'd be happy to take some of those.
Thank you for that, Andrew. We do have a question that was dropped in the chat asking how mouth microbiome DNA is passed between generations.
That's a great question, actually. So right now, we still don't really have a great idea as to how the microbiome within humans altogether first comes about. There's been a lot of discussion as to how do we get the bacteria in our small intestine, in our gut. The main reason because the bacteria somehow have to get there. If they actually can't get there through the oral cavity because the pH of the stomach usually will kill off a lot of bacteria that we find within the gut. So we're not actually sure how they got there. So there is thought to be some sort of a transmissive element between generations, I think mainly because you're in such close contact with them. What we've seen from studies of family units is that you share a lot of bacteria with the people that you're close with. You actually also share a lot of bacteria with pets too, which is kind of gross, but kind of cool. So I guess the short answer to that question is we are not 100% sure, but I do think that there is not only a kind of close contact relationship between individuals, so sharing of bacteria that way, And then I also think that your genome itself, the DNA within you, also contributes to the type of bacteria in your mouth. So that's something that I'm interested in looking at in the future.
Wow, that pet thing really got me as a cat owner. Does anyone have any other questions?
I'm also super happy that dire wolves are a real thing.
They are real. They are real.
Yes.
Hopefully that paper will come out within the next month or so, and you'll.
Now, did you actually have to just fully destroy that jawbone, or did you only take bits and pieces of it, or?
Yeah, I had to destroy it. It was so sad. I mean, it's so old. That's the thing, you know, with this field and with the field of archaeology too. It's one of the only things that you're actually destroying what you're trying to observe.
Wow.
Yeah. So with a lot of this stuff, and this is actually why dental calculus has been really kind of looked on as potentially the savior in the field, is because previously I had to go in, break that bone apart, and cut all the roots off, and try to get DNA from each of the roots of those teeth that you saw on that jawbone. With dental calculus, you don't have to do that. You just pick off some plaque. You don't have to damage the specimen at all. And actually the really big thing now that a lot of big labs in Europe have been investigating is you can take a sample of soil from where you know to be like next to a deceased, like some remains, not even the bones, just the soil next to it. And that's enough to actually get genomes for that individual, for the human, for that animal that you're looking at, just the soil. So,
Wow.
Pretty cool.
Yeah.
We have one more question asking why in particular urine is such a good source of study.
Yeah, we were kind of thinking about that as well. So what we compared to feces, it's amazing. And one of the big reasons, as I mentioned, is that feces has a lot of enzymatic activity, which breaks down those DNA basically fragments the DNA. So even if you collect the fecal material right away, you're getting a lot of damaged DNA, and you're getting 99.9% bacterial DNA from there, because there's a lot of bacteria in feces and in our guts. So what we generally think is happening is the urogenital system is generally considered to be pretty sterile. Urine is generally considered to be pretty sterile. So what we found is that what we're pulling the DNA from is shedded cells from within the urethra. So basically the genetic material, you're not getting really any bacterial contamination in urine. You're really only getting the cells, even though there's not that many, of the human or animal source in this case. So yeah, that's generally why we think urine is such a really nice indicator of genomes, or you can use it to find genomes, is because it's not contaminated, and because you actually get a lot of cells shed during urine too, it seems like.
Wow, who'd have thought? Does anybody else have any other questions? Feel free to put in the chat, Q&A section.
Yes, you can always email me too if you need to. It's aozga@nova.edu.
Still quite a few people in there. If you guys have questions, we can hang around for a couple more minutes. We'd love to. Andrew, it was really informative. I appreciate it. I know there's a lot of students and myself and Nicole and anybody that misses it today, we are going to record it and obviously publish it in the YouTube HCAS channel, and as well as the Shark Media as well. So, we can also get you a copy as well.
Great.
Might be it, Nicole. I don't see any other questions coming
in. I don't either.
That's okay.
We're stumped.
Sometimes there's a little bit of a delay, but so, all right. Well, Thank you, everybody. I mean, not a question, but a great presentation and comment on there as well. Research is really interesting. So there you go.
Thank you. Mm-hmm. All right. Yeah. Thank you both for hosting. Thank you all for attending. I really appreciate getting the word out about this stuff. It's something that I'm passionate about, and I'm going to keep going with it. So
awesome.
That's awesome. Thank you.
Thanks. Have a great night, guys.
That was a great presentation.
Thanks.
Bye, everyone.
Should 2 more still. One person's still there. There's one person still in there, Andrew, but I was just going to say thanks again. It was awesome.
Thanks. I appreciate that.
And even in the springtime too, I know you've already done one, but it's one of those things that, like, if you have some sort of research specific topic, or you want to even do something from where we can film from the lab, where you can show us demonstration pieces of fossils or whatever it may be that, like, you can actually show us too. We can figure that out and get you scheduled for the spring too, because a lot of the students, even from freshmen all the way on, have really a lot of interests and passions, and that's why they jumped into this. So I think we had 30 Joes. We think we had 19 tonight as well.
Awesome.
Some other faculty members too. So
great. Yeah, I'd be happy to. I mean, I think I'm probably going to be wrapping up the study at the end of next semester, so might be interesting to see what kind of bacteria are in.
Now, are you going to have any grad students on your?
No one has expressed interest in me yet. I haven't gotten contacted, but I also don't have a grant to pull in to pay one yet.
Gotcha.
So once I know Dr. Lopez has been pretty successful with that, so he's gotten a few grad students and a postdoc and stuff. So I'm trying to get the ball rolling with all this. So, yeah.
When you do, you're gonna have some. So I mean, I always do stuff super interesting. I think some students will be sending you some emails. So
thank you.
All
right, great guys. Thanks again, and that was fun.
Yep, we'll send you an email too as well, and some couple other follow-up stuff on it. So
awesome, great.
All right, have a good.
Thanks. You too. Should I end the whole meeting?
Yeah. Yep.
Okay. All right. See you guys.